Okay, so we talked a lot about proteins and kind of the different structures, be able to identify those structures for the test. So nucleic acids. Okay, let's talk about nucleic acids. The amino acid sequence of a polypeptide is programmed by a discrete unit of inheritance known as a gene. So therefore the definition of a gene is a discrete unit of inheritance. So genes consist of DNA, deoxyribonucleic acid, which is a type of nucleic acid. And so you inherit that from your parents, and that DNA is kind of the recipe book, the blueprint, if you will, for its own replication and creating proteins, if you remember the shape of the proteins. So DNA creates a different shaped proteins produce all the different phenotypes or things we see. So blue eyes, brown eyes, that's kind of thing. And so it directs the synthesis of proteins and directs the activities of the cell. But it doesn't do it directly. It has kind of this go-between called RNA, which is ribonucleic acid. So deoxy means it's a ribonucleic acid that's missing an oxygen. So RNA, ribonucleic acid. DNA is what's called transcribed into RNA. So when you talk about a scribe, someone who writes, okay, so it copies down the DNA, the RNA copies down the directions from the DNA, and then it is translated into proteins. Those directions are translated. So you have in there your DNA, your gene, then it is transcribed into RNA, and then the RNA is translated into your amino acid sequence. There's a polypeptide chain that then forms a protein, and the proteins do all the work. So they're composed of monomers called nucleotides, and nucleotides have 3 different parts. So 5-carbon sugar called ribose, hence ribonucleic acid, right? DNA deoxyribose, so that's 5-carbon sugar without the oxygen. A phosphate group and a nitrogenous base. Those are your A, G, C's, and T's. Okay, so phosphate group, your pentose sugar, your nitrogenous base. In this case, that's an adenine or an A. And so the different bases, this says A, T, C, G. Right? And so just how those line up, all the different ways to put those together, create all the diversity that we see, and that's just absolutely incredible to even think about. So RNA also has A, C, and G, but it has a T instead of a, but instead of a T, it has a U. Okay, so it'd be A, U, C, and G. And so the nucleic acid polymers. And the polynucleotide, it forms from the nucleotide monomers, so the single to the multiple. And so they bind together through dehydration reaction, and so that creates that repeating sugar phosphate backbone. So you get that double helix backbone, and you have your protruding nitrogenous bases in the middle, and so there you are. There's your backbone, and there's your bases. Okay, and that's put together by that dehydration synthesis. And so when you take them and put them together, it forms that double helix, and so each one of those bases pairs with another, and so A pairs with T and C pairs with G. And so you have, just think of at, trying to remember what pairs, and then you have C and G. And so in your RNA is usually a single strand, or your DNA is usually a double strand. So there's a double strand, and you see them pairing together, the ats and the CGs, they pair together, and those nitrogenous bases fit up, and they use hydrogen bonding. in the middle to make that pair. So back to our lactase. Most people stop producing lactase in the childhood, and makes it hard to digest milk. And so lactose tolerance is relatively recent mutation in the human genome, and it became advantageous for human cultures with dairy or access to dairy to keep this, so they could digest milk late into their adulthood. And so, and there's 3 mutations that keep the lactose gene permanently turned on. And it occurred about 7,000 years ago. So up until 7,000 years ago, almost everybody was lactose. I'm assuming maybe 1 or 2, but 99% of everybody was lactose intolerant. But about 7,000 years, coinciding with the same time as the domestication of cattle, we start to get lactose tolerant people into adulthood because of the use of cattle. And so that's a recent mutation, a recent evolution. Remember the change in allele frequencies over generations. So those allele frequencies changed, different genes, and now they can digest sugar. So that's the end of chapter 3. There's going to be some questions. Answer those, and let's move on. These EWE classes go fast, and so let's jam like jelly.